Okay, well, welcome everyone. My name is Jennifer Boyko. I'm the Senior Manager of Scientific Operations with the Canadian Longitudinal Study on Aging. Thank you for joining the CLSA webinar today that is entitled Metabolomic Sweet Spot Clock Predicts Mortality and Age-Related Diseases in the Canadian Longitudinal Study on Aging. Before we begin, I would like to acknowledge that the CLSA National Coordinating Center and McMaster University are both located on the traditional territories of the Mississauga and Haudenosaunee nations and within the lands protected by the Dish with One Spoon Wampum Agreement. The BC Cancer Centre in Surrey is located on the unceded traditional and ancestral shared territories of the Katzie, Kwantlen, Coquitlam, and Semiahmoo and Tsawwassen, sorry about that, First Nations. Simon Fraser University respectfully acknowledges the Musqueam, Squamish, Tsleil-Waututh, Katzie, Coquitlam, the Kikite, the Kwantlen, the Semiahmoo, and the Tsawwassen people on those whose unceded traditional territories our three campuses reside. As attendees of the webinar today, I encourage you to continue your learning following the webinar and to acknowledge the original inhabitants of the lands where we currently have the privilege to do research, live and work wherever that may be for you. Now let's review a couple of housekeeping points. Everyone but the presenters will be muted throughout the webinar. If you need to change or test your audio during the webinar, you can click on audio settings that's on the bottom of the Zoom window. At the end of today's session, there will be a question and answer period. If you have a question for the presenter during the webinar, you can post it in the Q&A box, which is located in the bottom toolbar. All the questions will be addressed at the very end of the webinar, and questions will be visible to all attendees. If you have any technical trouble concerning the webinar, please use the chat box to communicate with our webinar team. Finally, a feedback survey will be launched at the end of the webinar, and we do invite you to complete it after exiting the Zoom session today. The brief evaluation does provide us with important feedback that we can use to plan future webinars. Now for today's webinar, again entitled Metabolomic Sweet Spot Clock Predicts Mortality and Age-Related Diseases in the Canadian Longitudinal Study on Aging, presented by Olga Vishnakova, who is a postdoctoral fellow at the BC Cancer Research Institute and the Department of Biomedical Physiology and Kinesiology at Simon Fraser University. Dr. Vishnakova holds a Bachelor of Science and Master of Science degrees in Applied Mathematics, as well as a Master of Science in Computing Science from Radboud University and a PhD in Computational Biology from Simon Fraser University. Her research focuses on healthy aging using multi-omic approaches to study heterogeneity in aging and also the biological mechanisms underlying resilience. So welcome to our presenter as well as all of you today, and I look forward to a great session. Over to you.
Thank you for this warm introduction and for having me here today. And also for your interest in our work. I'll walk you through the challenges in the field that motivated this research, how we use untargeted metabolomic data from CLSA to overcome these challenges, and introduce the sweet spot clock, a molecular predictor of aging, to investigate how we age. We know that age is a universal process, yet each individual experiences it differently. Although chronological age is a major risk factor for many age-related diseases, cancer, diabetes, cardiovascular disease, still it does not consistently predict disease susceptibility, treatment response, or variability in aging trajectories. Research in aging investigate this variability on multiple scales. It may look at macro level, at the scale of a whole body, and here We may look at frailty, cognitive or physical function, and we use instruments such as frailty index, static load, or comorbidity index. Next, we may focus at a specific system and investigate variation in cardiovascular health, neurological function, or skeletal health. Molecular biology can look even deeper on a cellular resolution and look at cardiomyocyte senescence, microglial activation, or specific skeleton cell dysfunction. But going even deeper into a single cell resolution, we can look at difference in health at the level of a specific components of a cell organelles and examine the reason for mitochondrial dysfunction, energy plant cells, or lysosomal impairment compromising waste center of the cell. Finally, we may go as deep as at the micro level or omics and try to find the roots of these differences in aging patterns at the molecular level, starting from genetics or blueprint, and further down the road through DNA modification, gene expression, translation into protein and their degradation. And that allows us to investigate the genetic predisposition for specific disease or identify dysregulated pathway. So my talk and my research focuses on this bottom layer and on a high resolution scale. So what is aging? It's often described as time-dependent decline in function that results in accumulation of cellular damage over time. And it might manifest as through, for instance, mitochondrial dysfunction or genetic instability that contribute to cancer. But on the bright side, for some people, it's not the case. Some people experience an early onset of age-related diseases, while others remain remarkably healthy well into old age. And this heterogeneity of aging, and actually the main question, why do some people stay biologically younger? That's exactly what drives this research. So what is biological age? It might be described broadly as the level of age-dependent biological changes, such as molecular or cellular damage accumulation. But in aging research, we are doing way more simple calculation, and we treat it as a numerical value, which corresponds to the clinical age at which an average individual in a reference population displays similar age-related biological changes. So if we age faster than an average person in a population, we are seeing that this person is a faster aging, and vice versa. If we are age slower than an average one, so we're talking about slower agers. And the question is why we even bother of measuring this biological age. There are multiple reasons. One is really straightforward. We can quantify individual deviation from some normative aging patterns. How different the specific individual from an average person in a population. But we also can identify factors, including modifiable risk factors. Some of them are known and others still unknown, associated with accelerated or decelerated aging. And obviously, we also can identify some life course events that drive the divergence and aging trajectories. Ideally, with these biomarkers of biological age, we can assess the effect of longevity interventions. Just because, in contrast to some animal models, we cannot wait for 10, 15, 20 years in order to assess the quality of this intervention. So we do need some proxy for mortality in order to say how good we are in terms of specific clinical intervention. And the idea of measuring biological age on a molecular level has been gaining traction for over a decade, when the early attempt to build these biomarkers using telomere length until in 2013, Dr. Howard introduced his well-known palm tissue clock, and actually he coined the term biological clock. And since then, many other biological clocks using different omics data appeared, using data such as gene expression or proteomics or metabolomic. Basically, how these models work, so we are using some type of omic data, either proteomic abundance or metabolomic abundance or gene expression data, feed them into some complex or less complex model starting from multinomial regression or tree-based model, or then some neural networks in order to predict biological age. And even though the many models exist already, we still do have some challenges with the quality of these models. First of all, as all data-driven models suffer, we have a limited specificity, and we cannot answer a question: do these molecular biomarkers reflect true drivers of aging, or they also have some consequences of aging in it, or some unrelated signals just because they correlate with some true drivers? Some models, especially tree-based model or neural network-based model, have some problems with interpretability, just because we cannot clearly answer what individual feature represent. The other problem is, and the other challenge is the choice of a trained targets. We do have a third generation clock trained on chronological age. We do have a second generation clock trained on mortality. So the question is, what should serve as a primary training target to predict not just mortality, but a broad range of age-related outcomes? And we do have two other challenges in the field, such as nonlinearity, especially with multinomial regression models. How can we account for complex relationships to capture more realistic patterns, such as nonlinear patterns, while preserving interpretability of the model? And finally, we know that aging is multidimensional. So the question is, can a single omic layer, let's say epigenetics, capture it all? Or do we need many different omics in order to make a prediction? So in response to these challenges, we started to work on omics-based aging biomarkers with an ambitious aim to make it interpretable and focus on relevant features, account for nonlinearity, choose a target for training in such a way that we can predict not just mortality, but multiple age-related diseases. We also want to discover some dysregulated pathways and ideally use multi-omic data for these predictions. So to build this biomarker, where we use data from Canadian longitudinal study on aging, which provides really rich participant profiling with the genetic data, metabolomic data, DNA methylated data, and also rich phenotypes. We used obviously a comprehensive cohort with omics data available at the baseline. So first question that we asked was, can we identify measures relevant to health and healthy aging? And if we can we determine the optimal ranges so we can further account for non-linearity? And when we talk about health and healthy aging, we first should define what is it health. And there is no simple answer to that question. Otherwise, what we are doing here. So we used 5 different instruments to assess health that try to capture different aspects of health. First, we calculated the deficit accumulated frailty index, which is used 51 health deficit to represent overall health. After that, we focused on morbidities, and we split it into 2, and I'll explain further why we did it. So we first calculated as a normalized number of 5 major diseases: cancer, cardiovascular disease, major pulmonary disease, dementia, and diabetes, and complemented it with the 28 self-reported clinical diagnostic condition as instruments number 3. Also, we calculated cognitive and physical function scores to capture, again, different aspects of health. We normalize it to a unit interval. So we have a scores from 0 to 1 in order to make it possible to compare between this. And if you're interested, so here is our correlation between our instruments. So once we have this, we know that some person is more or less healthy than other one. We started to investigate how exactly we can identify this health-related phenotypes. And we used our observation from the super senior study. This study was launched in our lab more than 20 years ago. Super senior phenotype was defined as being 85 years and older and never been diagnosed with 5 major diseases that comprise our instrument number 2: cancer, cardiovascular disease, diabetes, dementia, and major pulmonary disease. That this almost 15 or even more years of recruitment resulted in 700 super seniors, aged 85 to 110, recruited in BC. And these are physically and cognitive high functional group. So here is just some means for common tests for elderly people, such as time up and go or mini mental state exam. What we observe in this remarkable group, that our initial hypothesis was that they should have a telomere length, this protective cap on a chromosome, longer than an average population. However, no difference in mean were observed between 2 groups, but what we did see in reduced variation in telomere length compared to midlife controls. Also, similar patterns were observed for other hematology data, such as red blood cell count, hemoglobin, or red cell distribution with no mean difference, but difference in variance. So we used this idea in order to generate what a simple upon first glance hypothesis that measures that are important for health tend to be closer to some optimal values or sweet spots among the healthiest individuals. What's important here that by measuring the distance from the sweet spot, we can quantify the level of dysregulation and inform dysregulated processes. And this deviation from sweet spots also can serve as nonlinear transformation when we are talking about some complex model down the road. This idea was formalized and two-step statistical approach where we first screening phenotypes of interest where variance heterogeneity across health extremes, meaning that we compare variance between most healthy and least healthy groups. And after that, we estimate optimal phenotypic values where deviation from these values link to poor health. And we are doing this using segmented regression with a 3-node point. Applying this framework to phenotypic data and available in CLSA, we were able to identify more than 100 of phenotypes with significantly higher variance among the least healthy. Why we started from phenotypes, but not from more complex metabolomic data, because first of all, we wanted to validate this idea and understand whether statistics can help us to identify related features where we do not introduce any prior biological knowledge. So our results suggest that indeed we can identify health-related phenotypes that make sense just because we know that they make sense. When the hemoglobin A1C, an indicator of an average blood sugar that show the strongest evidence of difference variance across both sexes and all age groups. Also, majority of these phenotypes appeared to be related to health in both sexes. Some of them unique for either for females or males. Another key finding that was observed from the first step of this study is that about 40% of the phenotypes of interest exhibit the sweet spots. And here is just one example for preteroxine, how we can determine that a specific phenotype has a sweet spot. And another important observation that confidence intervals for identified sweet spots appear to be narrower than a standard clinical reference interval. Here is just one example. The confidence interval for heterogeneity identified with the frailty index as a health instrument compared to a reference through the reference interval. What we did next, we cross-checked identified optimal ranges with the clinical reference intervals used in a light lab or Surrey Memorial Hospital lab. And for majority of blood chemistry biomarkers, we did have a match with an exception to cholesterol and HDL. One of the reasons for that is that we probably should be more. We do know that there are many CLSA participants all on statin. probably that disrupt the analysis and identification, as we do not see a clear picture of the true distribution of these phenotypes. So with that, we are moving forward, and we ask ourselves, how does the metabolome reflect variation in aging across individuals? First, couple of words, why metabolomics is so important and what is it? Metabolome is a collection of all metabolites, which are small molecules in the body, and metabolome is the end product of metabolism, and it is the end result of all environmental and biological processes, starting from protein degradation, medication intake, our nutrition patterns, how we exercise, whether we do have any exposure from some chemicals or even some exposure, whether we're exposed to some level of stress, what are our sleeping patterns, do we sleep well? All of these impact our metabolome. What is also important that about 30% of identified genetic disorders involve defects in small molecular metabolism. And this metabolite, these small molecules, act as cofactors and signal molecules for thousands of proteins. Given its importance, we did start from metabolomic data available in CLSA to calculate this metabolomic biomarker. If we talk about CLSA, so we do have data for almost 10,000 participants at the baseline. We specifically focus at endogenous metabolites produced by our body that might be kind of categorized, metabolites might be categorized based on the super pathways, with a majority of being liquids, lipids, and some smaller groups such as amino acids, cofactors, and vitamins, nucleotides, and so on. We are restricted to our analysis to metabolites with less than 20% missing rate, and that resulted in this nice number, 88 metabolites for further analysis. So we applied the statistical framework I was talking previously to this complex metabolomic data, and we were able to identify 178 or 20%, that 20% of metabolites showed significantly higher variance in at least healthy individuals. So we looked at the top biomarkers, and they completely make sense. For instance, N6 and N6 metallolysis as a derivative of amino acid lysine, and it's a component of histone protein. Or the second hit was glucose. Obviously, we all know how glucose is the fuel important to our body. And these health-related metabolites, all 178 of them, were used as a predictor of metabolomic age. Next, we wanted to account for nonlinearity, so we calculated sweet spots. So we determined optimal level. We're able to do that for 74 metabolites out of these 178. What the remarkable result here that more than half of them, 95% confidence intervals for identified sweet spots, did not include population mean. So we assume that that might be partly because the older Canadian population might, and it might be quite a strong claim, not healthy enough in order to maintain an optimal value of a specific optimal boundaries of a specific metabolite. What we did with this many, many small molecules and the abundance, again, they were used as an input to a model for those metabolites that exhibit the sweet spot. We performed a transformation, specifically we calculated the Euclidean distance between a sweet spot and measured metabolomic abundance in a specific person, and either transformed or untransformed, where we do not see any nonlinear patterns, where after that fit to plastic net, which is a double penalized regression, multinomial regression model, in order to predict frailty index. While frailty, again, we didn't want to use mortality as an outcome because we did want to see changes not directly linked to the later in life changes. We want our biomarker to be predictive to multiple conditions, not just mortality. So we chose some composed representation of health status. And here what were our results are. So we basically compared this biomarkers against some baselines. So we first were interested whether this transformation makes sense. We also wanted to know whether choice of target is right. And we obviously compared with the frailty index itself. And obviously, so this results are on a train. So we split in a train and test set, made all the training on a training set, and assess the quality of the model on a test set. It's pretty small, so it's 1,200 of participants. So that's why probably you won't see any extremely strong association with mortality. But nevertheless, the model and how it was built definitely makes sense. What we did after that, we calculated this. We assessed biological age, and here is a regression. So we regressed onto age. Above the regression line, we're talking about participants, CLSA participants that age faster than average, and below are slower agers. What we also observed is that metabolomic age deviation, these differences with higher amount females rather than males. Next, we measured association with all-cause mortality and age-related disease between this molecular metabolomic molecular biomarker and diabetes, cardiovascular, so represented by strokes, kidney failure, Alzheimer, cancer, and pulmonary diseases. We did observe strong association with mortality even in our smaller test set, but not for Alzheimer's and cancer. And again, there is a reason for that. Dementia was an exclusion criteria at the time of recruitment, and metabolomic data were available only as a baseline. So we did not expect to have to see much for Alzheimer's. Same for cancer, it's too heterogeneous with too many, with just a few for each type in a test set. So that's pretty predictable. So in order to understand what does this mean, so what does this mean to have a hazard ratio of 1.08 for all-cause mortality? So what we did, just to get an intuition behind that, was split participants into four equal parts, and compare the survival probability for each of them for slowest agers and for fastest aging. And now it kind of Now we feel what does this mean. And after that, we went even further and we looked at two extremes, fastest and slower agers. And here the difference in survival even more pronounced. So obviously we measured association with health, lifestyle, and socioeconomic factors, and we observe expected association and expected direction. Obviously, nutrition, smoking, alcohol consumption, all of this contributes to faster aging, while income and level of education reduce our biological age. So what we also wanted to know whether our metabolites that we used as a predictors, as it's a data-driven model, so it's made some feature selection during the training process. So we will wonder whether these features remain after selection make sense, biological sense. So what we did, we used 50 top predictive metabolites selected by the model and looked at association between them and 14 known disease biomarker, chronic condition, and diagnostic measures, so all what we can find could find at that point. And all of these metabolites showed strong association with at least 5 distinct phenotypes after multiple testing correction. Here is just a piece of the results, not suggesting to look over, but just to explain that indeed each of these metabolites makes total sense. And it's associated with multiple diagnosed conditions. We also performed a validation of this result in super seniors, and for that we performed a metabolomic profiling for super seniors and age match control exactly the same way as it was done for CLSA data at Metabolon with the same control samples, just to be as close for validation as possible. And that resulted in a positive results, obviously. So we did observe that biomarker trained on CLSA data and applied to super senior data set was able to catch a difference between healthy and age match control, so extremely healthy individuals in terms of phenotype and age match control, and was significantly associated with mortality. What we also wanted to look is where our metabolomic biomarker stands compared to well-known epigenetic, highly promoted epigenetic biomarkers. Hahnemann and Horvath plots are available at a baseline as a part of comprehensive cohort data set. We also calculated state-of-the-art GrimH boost version and PinoH using BioLearn Python library generally provided by Biomarkers of Aging Consortium. And what we observed is that our sweet spot clock based on metabolomic age deviation outperform epigenetic biomarkers and was stronger associated with mortality. I obviously acknowledge that GrimH, PinoH, and Hahnemann Horvath clock were trained on a different data set that I believe US-based participants, while a sweet spot clock was trained on even on a separate training set, but still on a CLSA. So we expected to for this metabolic biomarker to perform better. Yes, there is definitely some bias, but nevertheless, remarkably better, which is good. What we wanted to know, and I already expressed that we want to know that whether multiple layers capture unique, whether each layer capture unique biology or not. So for that, we actually built a multinomial model and used our biomarker and image as a predictor, and that actually improved the performance of the model measured by a concordance index. So indeed, different layers capture different biology. So we obviously ask ourselves whether, first of all, whether we can do better than pre-image in terms of developing biomarkers, and whether these two epigenetic based and metabolomic based share something in common. A couple of words about epigenetic, we still have time. So it's a heritable changes in gene expression without altering DNA sequence. And if we, in simple words, how we can think about epigenetic, we do have a multiple cell types. We do have a muscle cells, we do have our immune cells, all of them cells, but they express different, but each of them have a different gene expression. And that is with the help of this methylation protein that either silence some gene expression or allow them to be expressed. And the problem is that epigenetic is shaped by environmental development and aging, and it also might be impacted by our habits such as smoking. So the question is whether we can use this DNA methylation pattern in order to build these biomarkers. And again, we looked at two different level of changes, such as different differentially methylated position across the genome and differentially variable position across the genome, and aggregated into regions, and that resulted in epigenetic biomarkers. What we also observe is that there is almost a little overlap between these two. And using this information, we were able to build with a quite a fancy model epigenetic biomarkers where we first predicted health status, and after that we weighted this prediction using mortality data. It makes sense. So we show that this model makes sense. We compare it against benchmarked models. What is relevant to today's talk is that there is just a moderate correlation with metabolomic and epigenetic biomarkers. Each of them has a genetic component. Each of them might be partly explained by known lifestyle, socioeconomic factors. However, each of them still has this large portion of unknown, which is still need to be investigated. So the main takeaway is that we proposed methods that allow to identify measures related to health and healthy aging, and by measuring deviation from them, we can quantify the level of dysregulation. We also show that our omics biomarkers are predictive for mortality and onset of age-related diseases. Talking about utility, and the major clinical implication is that using this or similar models, we can create individualized health report cards that pinpoint dysregulation pathways and able to predict future health risks, complementing genetic risk score. Now, we also with help of these biomarkers that may help guide lifestyle and prevention efforts. And moving forward, we can investigate how lifestyle or pharmacological intervention can modulate biological age deviation and integrate multiple omics and a single by maybe ensembling single omics prediction, which is the easiest way we can improve the prediction ability of such biomarkers. And that would wrap up my presentation. With this nice comparison, I love to think about healthy aging as a magic cocktail, and unfortunately, we just don't know its recipe. So I want to believe that our study made us tiny bit closer to the secret of this recipe. We just need to dig deeper. And with that, I want to acknowledge both of my labs, my amazing supervisors, lab members, collaborators, and obviously our participants. Without them, this research would not be possible. And thank you for listening.
Great. Well, thank you very much, Dr. Vishnyakova. We will go on to some questions now in the Q&A. Some of I know the first question, I pre-read it, is long, so I'm going to read it out loud, and maybe you can also read along and then reply to Walter. Really interesting research. Thanks for sharing. In the associations of metabolomic age deviation with age-related disease and all-cause mortality on slide 34, the odds of the individual disease outcomes, such as diabetes, stroke, etc., were relatively similar. Would I interpret this to mean that, would I interpret this to mean that metabolomic age deviation is a precursor of disease and death, but the specificity regarding risks for specific conditions is still unknown?
I'm just double checking that. So, and it's not exactly the same, but similar, partly because we incorporated metabolites related to each of these diseases, just because we trained on frailty index and frailty index already include markers, not markers actually, but health deficits such as whether a person was diagnosed for diabetes, for cardiovascular, so on, and it was treated equally in frailty index. So we included metabolites related to each of the diseases as a predictor of metabolomic age. Yes, in terms of an answer to this question, it's not specific to a single disease, but we're still able to catch dysregulation leading to each of these diseases. I hope that it just...
And the next question, fascinating work. If I interpreted correctly, you observed a sex difference in the distribution of the sweet spot clock. Can you comment on what may be responsible for this?
Obviously. So indeed, we performed all this analysis stratified by sex. So we acknowledge that adjusting for sex would not result in a proper estimation. Sex biology works differently. And we expected to have a hormone level metabolites that are precursor to sex hormones to have different optimal levels between sexes. So that's totally makes sense. Yeah, that's an answer.
Okay, and if there's any more questions, just a reminder, if anybody does have any more questions, to type it into the Q&A box. But for now, we have this last one. Could you please recap the calculation of the sweet spot?
Sure. So it's a two-step statistical framework. So first, we'll look at difference in variance using Brown-Forsyth test for heteroscedasticity. We looked at two extremes and statistically tested the difference in variance. And after that, the metabolites that pass multiple testing correction after the first step, from them we tried to apply the segmented regression model with a single node point and looked whether we observed any change of trend. And if we did, we looked at the direction of changes. If direction was consistent with our expectation, we treated this node point as a sweet spot.
Okay, well, I think we will slowly start to wrap up then. I'm sure if anybody has any other questions, they can reach out to you directly. But for now, I'd just like to thank you very much. And I also would like to do an extra special thanks because I know you jumped in at the last minute to do this webinar. So much appreciated. But I'd now like to remind everyone that the next deadline for data access applications, if you're interested in doing your own research, is July 8th, 2026. Please visit the data access section of our website to review what data is available, as well as additional details about the application process. Also, a reminder for all CLSA data users to keep us updated on any changes that may affect your data access agreements. Have you moved institutions, or has your research team changed, or has a trainee graduated or moved to a new position? You can always reach out to us at access@CLSA-ELCV.ca with any questions or updates. I'd also like to remind everyone to complete your anonymous survey upon exiting the session today. The CLSA has introduced an updated data access fee structure effective October 2nd, 2025. The first adjustment since we actually launched the platform more than a decade ago. This update supports a sustainable cost recovery model that will help us maintain exceptional quality and stewardship of CLSA data for years to come. And if you'd like more information about this, please visit our data access webpage on our website, or you can click on the link that's provided in the chat. And lastly, about our upcoming webinar, the details of our next webinar are still being finalized, but you can visit our website under webinars to stay up to date on the next webinar. If you or a colleague are interested in presenting a CLSA webinar, please reach out to us on our webinar team. We would be glad to hear from you. And the recording from today's webinar and the slides will be available in the coming days on our website. Finally, you're invited to fill out the exit survey that will appear right at the end of this webinar. And thank you all for your attendance today. And again, thank you to Dr. Vishnyakova. Bye, everyone.